Everything you know is living is made of cells. Every eukaryotic cell makes up more complex organisms, such as this penguin, and is controlled and constructed by DNA. Proteins, an integral part of the function of a cell, is constructed from the DNA through the process of protein synthesis. DNA is a unique and complex molecule. To transcribe, DNA unwinds from its double helix shape to allow itself to communicate instructions from the nucleus through messenger RNA. A segment of DNA, commonly referred to as a gene, acts as the template for the making of mRNA. The template DNA will unzip through the enzyme helicase. This allows the mRNA to make a copy of the DNA by forming together nucleotides that are complementary to the DNA's single strand of nucleotides. If the mRNA forms from the leading DNA strand, 5'3", polymerase reads with no breaks and everything is peachy. If the mRNA forms from the lagging DNA strand, 5'3", then the polymerase has to work backwards in Okazaki fragments. After mRNA is formed, RNA polymerase rejoins the DNA strands, and mRNA is released to float out of the nucleus through the nuclear pore to the cytoplasm with instructions from the DNA molecule. The mRNA strand travels to a ribosome embedded in the endoplasmic reticulum, or is embedded in a free-floating polyribosome in the cytoplasm. The ribosome, made of both the small and large RNA subunits, accepts the mRNA into its S site. tRNA, free-floating in the cytoplasm, comes over to the ribosome. Initiation. What follows is known as translation. The tRNA attaches to the mRNA by aligning its anticodon sign with complementary nucleotides to the codon of the mRNA. tRNA are incredibly specific to the codon on the mRNA. The tRNA then helps translate the message from the mRNA into an amino acid. The codon, with its 3 amino acids, tells the ribosome which amino acids to form. For the first section, the first amino acid form will be methionine, coming from the codon GUA. Once formed, the amino acid, its tRNA, and the string of mRNA move over to the P site. Another tRNA takes up the A site and encodes another codon into an amino acid. The amino acid from the first tRNA detaches from its tRNA and binds to the other amino acid through dehydration synthesis. The bond that forms between the 2 amino acids becomes a peptide bond. Now the dipeptide and the tRNA shift over from the A site into the P site, allowing more tRNA to enter the ribosome. Elongation. This translation of codons continues unendingly until it ends. Termination. Once all of the mRNA codons have been translated into amino acids and bound together by dehydration synthesis, the final codon signals the amino acid sequence to stop. The polypeptide then floats off into the rest of the cytoplasm, now a primary structure protein. This protein is non-functional until it is sent to the Golgi body. The process then starts over for any new protein that needs to be formed. Thank you for watching. This production is made by Eden and Meriwether. Yeehaw!